Hi guys, so today's topic is stem cells. So what are stem cells? Well, the human embryonic stem cells can be maintained in tissue culture as an immortal cell lines with the potential to differentiate into any cell in the body or by incorporation into the blastocyst to give rise to a new individual. So a stem cell can differentiate into any cell. Adult stem cells are present in every tissue and may be manipulated to differentiate into a range of cell types. These cells may be isolated, multiplied in culture, and then used as homografts to treat problems in their donor without risk of immune rejection or ethical concern. So let us talk about the properties of the stem cells. See, the stem cells are potentially immortal due to the asymmetric division. Stem cells are found in all the tissues. It occupies a niche where their numbers are regulated to remain relatively constant throughout the life. Surplus stem cells may undergo apoptosis so as to retain the appropriate number. So, it may be activated to undergo mitosis to support tissue growth or repair or maintenance. Otherwise, its number is maintained to an appropriate number. Others may go apoptosis, so it is regulated to a relatively constant number. Asymmetric division, one daughter cell remains undifferentiated and potentially immortal. One need to understand this, that asymmetric division, one daughter cell remains undifferentiated and potentially immortal. Differentiated phenotypes is strongly influenced by the environmental signals. Stem cells do not express the connexins or form the gap junctions. This is a very important point, that stem cells do not express the connexins or form the gap junctions. So stem cells niche. The stem cell function depends on maintaining a stable population of stem cells, which may be activated when the required and whose daughter cells can gain access to appropriate regions of the tissue. So whenever there is a wear and tear, so these stem cells get activated only when required. This requires a tissue niche where stem cells are grouped for control of their self-renewal and differentiation. So specificity of the niche environment depends on the paracrine signaling from the supporting cells, formation of the gap junctions with supporting cells, and on localized extracellular matrix molecules such as heparin sulfate proteoglycans HSPG4, which bind cytokines, fibroblast growth factors FGF, and other signaling molecules. So how is the activation of the stem cells in adult tissue take place? Well, the maintenance of an adult tissue requires a perfect balance between the cell death and the production of the newly replaced cells. So the stem cells are activated under the different circumstances of the tissue maintenance or for a tissue repair. The tissue maintenance is the simplest case, employs local feedback system where the differentiated cell signals stem cells to inhibit their proliferation. So when the differentiation occurs from the daughter cells, these differentiated cells only signals the stem cells to stop dividing and stop making other differentiated cells. So activation of the tissue repair may be reflect the exposure of the heparin binding growth factors such as the FGF2 signals from the macrophages as a part of the inflammatory reaction, and the paracrine signals from the damaged cells. Now talk about the specificity of the stem cells differentiation. See, the stem cells differentiation within healthy adult tissue is tissue specific, and new cells are appropriate in type, location, and number. Human neural stem cells are allowed to aggregate with human myoblast, differentiate into myoblast. Embryonic human endothelial cells co-cultured with the cardiomyoblast can differentiate into cardiac myocytes. A single stem cell population may generate several cell types, as seen in the hemopoiesis in bone marrow or in the basal layer of the epidermis. We can see that a single cell differentiates into so many different cells. In the epidermis, stem cells may differentiate into sebaceous gland cells, hair follicles, and keratinocytes. So single stem cells differentiate into different stem cells and the expression of the connexins. See, the connexins are the family of the proteins which form the gap junctions. So it permits the movement of the ions and small molecules between the cytoplasm of the adjacent cells. The expression of the connexins is often used as a marker to define the transition between the stem cell and the differentiated progeny. So in stem cells, connexins are absent, but in the differentiated progeny, the connexins are present. What is the work of the connexins? It basically it forms the gap junctions for the it permits the movement of the ions and the small molecules between the cytoplasm of the adjacent cells. One more thing needed to be understood: the lack of expression of the connexins is also considered to be evidence of the malignancy in the neoplastic cells. So connexins are also absent in the neoplastic cells. The stem cells hierarchies here. So the stem cells are traditionally be classified in hierarchies. The fertilized egg and its progeny form the first three cell divisions are the totipotent. What? The fertilized egg and its progeny from the first three cell divisions are totipotent. Human pluripotent embryonic stem cell lines isolated either from the blastocyst inner cell masses or from the fetal testis or ovary appear to have similar properties, including the potential to give rise to a new human being, but it is not ethically approved. For ethical reasons, none has yet been permitted to express this potential. Pluripotent cells may be isolated from the adult tissue, a possibility that escapes the ethical problem associated with the embryonic tissue as well as the problem of the immune rejection. So it can be taken from the adult tissue. Stem cells within different level of their hierarchies have different levels of competence to respond to growth signals. So what is the carcinogenetic potential of the stem cells? The stem cells have the capacity to generate large number of progeny cells and share with the malignant carcinoma cells properties such as expression of the telomerase and lack of the expression of the connexins. So what are the properties? The expression of the telomerase and lack of the expression of the connexins. These are two properties same as of the cancer. So the stem cell theory of cancer suggests that many or most cancer arise from the defect in the stem cell growth control. So whenever there is a theory that whenever there is a stem cell growth control, there is a theory behind the cancer. So the normal cells respond to growth controls and apoptotic signals. What need to be understood? The normal cells respond to growth controls and the apoptotic signals are competent to differentiate and respond adaptively to environmental condition, eventually become senescent. So whenever in a stem cell, whenever there is a differentiated cell, it is signaling to stop producing more cells, differentiated cells, the stem cells stop. But in a malignancy, this doesn't happen. It keeps on producing. So stem cells and their derivatives are after disaggregation and grafting into a new environment appears generally to behave as normal cells in these regards, but if they're implanted as a cell mass, they may be less able to respond to local growth control signals. So a pluripotent stem cell may be isolated from the following adult tissue: the bone marrow cells, the human adipose tissue, the olfactory bulb, the hair follicle cells. From where we can take the stem cells in adult tissue? The bone marrow cells, the human adipose tissue, the olfactory bulb, the hair follicle cells. So nerves and stem cell activation. See, human taste buds are continually shed from the tongue and replaced by the proliferation and differentiation of the basal cells. This process depends on the integrity of the sensory innervation of the tongue. So there is a connection between the nerves and the stem cell activation. So when we see that following the partial hepatectomy, the proliferation of the hepatocytes is impaired if the vagus nerve is sectioned. So whenever we section the vagus nerve, the proliferation of the hepatocytes is impaired. So this shows there is some connection between the nerves and the stem cell activation. If skin is denervated, the epidermis is thinned and the keratinocytes proliferation is reduced. So this also gives a indication. So examples of the stem cells used to promote the tissue regeneration. In the olfactory system, the mammalian olfactory system has the distinction of being the only part of the central nervous system undergoing continuous renewal. So the olfactory system is the only system, mammalian olfactory system is the only system which goes on continuous renewal. Olfactory receptor neurons have half-life of few weeks, and after renewal of division of stem cells in the olfactory epithelium, which give rise to both neurons and to the supporting cells. Their regeneration is promoted by the growth factor secreted by the olfactory ensheathing cells (OEC) located beneath the olfactory epithelium. So the olfactory ensheathing cells are a form of glial, and there is a much interest in their possible therapeutic use to stimulate axon regeneration in other regions of the brain. Not only are the olfactory receptor neurons continually renewed, but also their target neurons in the olfactory bulb. So other uses of stem cells: use of chondrocytes for tissue repair. The articular cartilage has a low capacity for repair. So the mesenchymal stem cells with the capacity to form cartilage, bone and adipose tissue can be extracted from it. Human nasal septum is a good source for chondrogenic cells with potential to use to engineer transplant for the restorative surgery of the otolaryngology. So nasal septum can also be used as a good source for the chondrogenic cells. Now the regeneration of the adult teeth. See, development of the teeth depends on the epithelial mesenchymal interaction, which employs a higher conserved genetic signaling mechanism common to the formation of many other tissues. Embryonic tooth morphogenesis critically depend on the migration of the cranial neural crest cells into the first brachial arch. So their odontogenic potential is realized in response to signaling molecules from the oral epithelium. Adult human odontogenic stem cells have been isolated from the dental pulp tissue. So here we can see how from a teeth, the dental pulp tissue, the DPSC, these cells are taken and the non-neurosphere formation is occurring, and from that the Schwann cells, the astrocytes, oligodendrocytes, and these cells are being produced, and from the neurosphere formation the functional neurons are being produced. Now regeneration of the skin. The autografts of the cultured epithelial cells are commonly used to treat the burn patients. Several weeks are required to amplify cells take from the patient, and then grafted skin does not include the hair follicles. This is the problem. But to create a skin equivalent tissue suitable for grafting, organotypic co-culture of the keratinocyte together with the skin fibroblast offers a very good promise. So cochlear hair cells. Now we talk about the cochlear hair cells. See, the normal hearing depends on the integrity of around 3,500 inner hair cells in the each cochlea. We know that the cochlea, there are the outer hair cells and the inner hair cells. These cells are generated early in the embryo during the first trimester. After that time, they are not normally capable of renewal or regeneration and must survive for the individual's entire life because they are not regenerated. So they have to survive for entire life for a person to hear normally. So hair cell loss is the most common cause of deafness and is currently irreversible. In mammals, the auditory epithelium includes the differentiated hair cells with an orderly spacing among the supporting cells, similar in many respects to other sensory organs, in which receptor cells are degenerated from the supporting cells. So now here we can see how the embryonic stem cells, the pluripotent cell, differentiate into multipotent cells, the mesenchymal stem cell, the neural stem cell, and the intestinal stem cells. So here we can see this is the cochlea. Therapeutic use of embryonic stem cell isolated from the human embryos poses immunological and ethical concerns. As with any other organ transplant, There is a risk of tissue rejection. There is a public resistance to use of cells or tissue from the aborted fetuses or to collecting fertilized ova specifically to generate cells for transplants. Isolation of embryonic stem cells from the placental cord blood or the use of the universal human embryonic stem cell lines may alleviate ethical concerns. Example of the potential therapeutic use of the cell engineered to support regeneration are like the olfactory and sheathing cells to promote axonal regeneration across the spinal cord lesions. Engineered expression of the bone morphogenetic proteins or the sonic hedgehog by the periosteal stem cells to promote the cartilage regeneration. Use of the muscle derived stem cells engineered to express bone morphogenetic protein or the vascular endothelial growth factors to promote the bone healing. So examples of the stem cells used to promote a tissue regeneration are the reconstruction of the olfactory epithelium, use of the chondrocyte for the cartilage repair, use of the odontogenic stem cells to regenerate the adult teeth, regeneration of the dermis using the epidermal stem cells expanding in culture, restoration of the hearing by regeneration of the hearing cells in the organ of cochlea. Examples of the stem cells used to promote organ replacement. Well, reconstruction of the hemopoietic tissue, the endocrine cells in diffusion chambers to treat the diabetes, mellitus, and other endocrine dysfunction. Use of the stem cells to treat the genetic disorder like a dystrophy and Duchenne muscular dystrophy and the hemopoietic disorders like restoration of the immune competence. Thank you.